Shall we wait for 2 more minutes, Siddhant, or shall we start?
We should start, sir.
We'll start.
Yeah.
Good evening and a very warm welcome to everybody. At the outset, I would like to thank Indian Society of Oncology, Dr. Ramesh Billimaga, Dr. Toprani, and Dr. Arvind Krishnamurthy for such a nice initiative and endeavor. So this is the initiation of ISO, Indian Society of Oncology Academics. It's first of its program, and every month or every, at least 6 in a calendar year, we intend to do. We have few more lined up. In February, the radiation oncologists will be doing a workshop, an online workshop on stereotactic body radiotherapy. In March, there will be a surgical workshop led by Dr. Vijay Kumar. And in March, April, there will be another one by Dr. Hemant Malhotra. So there's a lot of academics coming up. This was in discussion in the ISO, in the ISO Executive Committee, that it is high time that ISO took active interest in academics because the membership have been increasing, and ISO conducted a very successful, an awesome conference in Chennai recently. So with all these in the backdrop, we decided as ISO that academics is the core of ISO, and ISO is completely committed to it. So without much ado, let me hand over to Dr. Richa Chauhan. I must congratulate Dr. Damodar and Dr. Richa for formulating such a wonderful program. Molecular biology is not, which is often heard by medical oncologists, keep hearing about it, but the other specialties, they do not often hear it. So there is a lot of interest, and I learned from Rajesh that there have been a lot of registrations. So I hand over to Richa, Dr. Richa, to introduce the program and take things forward. Thank you.
Thank you, sir, and thank you, ISO, for providing us the opportunity. See, we all understand that, cancer is in essence a genetic disease, right? Its development, its progression, the response to therapy, and also the outcome is just a reflection of the molecular alterations which has accumulated in the genome. And in the last few years, with the, lot of information from the human genome sequence and a lot of new DNA sequencing technologies which has emerged, we do have a lot of information. And now we all recognize that molecular biology is an important component across all malignancies, and the field of oncology is transforming from the cellular to the molecular level. So we thought it's an apt time as more and more people, especially the young oncologists, will learn about molecular biology, and they will start utilizing the new, techniques like the NGS and all, which is not very simple. It's actually complicated in their day-to-day practice for the way how they treat the patient. So we thought it's an apt time to have a focused webinar on the same topic. And I thank all the faculties, all the clinicians, all the oncologists, the molecular pathologists, and also the scientists who have given their time. And so without wasting much time, I think we should start the webinar, and I request our chairperson, Dr. Rajendra Toprani, sir, to kindly take forward the webinar.
Thank you, Kausav and Richa. So this was an initiative of ISO, as you all mentioned, and the topic that you have selected is also of great significance as going forward. It is going to be incorporated into our day-to-day clinical practice also. So without much time wasting, let me introduce you. The first speaker is Dr. Damodar Gupta. He is a senior scientist at Institute of Nuclear Medicine and Allied Sciences at New Delhi, and he's one of the EC member at ISO. And he's going to talk on NGS basics for oncologists. Following him, the second speaker would be Dr. Gauhar Shafi. He is from Mumbai, and he is the Chief Informatics Officer at OneSir Diagnostics India Private Limited. So he will be talking on workflow and interpretation of NGS. So let me invite Dr. Damodar to deliver his talk. Dr. Damodar.
Thank you so much for kind introduction, sir. And can you see all my slides now? Am I audible?
Yes, sir. Yes, sir. You are.
And
Yes, yes,
slides are visible.
absolutely.
Okay. So the topic given to me is next-gen sequencing for oncologist. I'm going to cover most of the basics that how next-gen sequencing is really important in case of nucleotide or any kind of alteration within the genome or genome products. So as you all know about the hierarchy of DNA from nucleosomes to double helical structure of DNA, which runs in anti-parallel direction, and they form a hydrogen bond between each other. And based on this hydrogen bonding, either it is between adenine to thymine or guanine to cytosine, all these alteration or any changes between the tautomerism or anomerism within the nitrogenous bases, it may be responsible for kind of a mutation, and that needs to be identified in case of ailment or cancer. So this is what very important, that when a gene is mutated or a nucleotide change is there, the complete product is changed. So when we talk about gene product, either from DNA to DNA or DNA to its RNA, or we can say it is mRNA, messenger RNA that contains different codons, which may be responsible, which may denote a particular type of amino acids. So the product of gene is mRNA or different type of RNA. But from mRNA, we consider that a polynucleotide chain is synthesized. Poly amino acid or amino acid chain is synthesized. This amino acid means proteins are synthesized. So when we talk about a particular protein, it is encoded from a particular codons, and any changes in these codons are responsible for change in amino acid or replacement of particular amino acid. And it may be that protein or that product may be accumulated in the cell or may be oncogenic in nature or help in cells for their transformation or responsible for transformation of a particular type of cell. Let me give an example. Suppose a gene product, it is mRNA, and when we talk about introns and exons which are responsible for expression of protein, that expressing zone, whereas introns, they get spliced out from the mRNA, and a particular mRNA, if we look on this particular M, if mutation is there and if M is replaced by B, suppose any codon is there, it make and back, they both are near about same. It means when we consider about a word meaning, it is going to denote same kind of amino acid, and a product that is going to form, it is going to be a particular cake. So Emily makes a red choco cake every day. It means that a cake is going to be formed. But when we talk about mutation or replacement of a particular nucleotide in a sequence, suppose A is replaced by O. So the meaning or the product is completely changed. And if it gets accumulated or if it is responsible for transformation of that particular type of a cell, then we may find a particular element in that particular organism, either it is somatic or at the inheritance level also. So when we talk about mutation in a particular genome or sequence, we can say either it is synonymous, it means that it is going to produce same encode for similar kind of amino acid. When we talk about non-synonymous, suppose A is replaced by T, it is going to indicate a histidine protein. This particular codon is responsible for or shows histidine protein, a histidine amino acid. So the protein structure is completely changed. So all these nucleotide replacement, addition, or deletion of a particular nucleotide from a genome sequence or from a gene may encode different kind of proteins, or it may lead to kind of a mutation which is responsible for transformation of a cell. So how to identify all these things? So Frederick Sanger, he initiated first that how to identify or sequence DNA using particular nucleotide or change nucleotide by dideoxysequencing method. So as we know that the deoxyribose sugar, it contains hydroxyl radical, a hydroxyl and 5'P when we talk about the DNA chain or DNA double helical structure, it runs in antiparallel direction. 5' prime end, 5' end is responsible, or we can say it is phosphate end, as well as the 3' end, it is hydroxyl end. So if it is replaced by simply hydrogen, then a termination or extension of a DNA or replication will be stopped. So when we remove this particular OH group from the deoxyribose sugar, the termination will occur. So incorporation of a particular nucleotide along with the fluorescent, if you take this particular zone with the fluorophore, we can easily identify a particular type of sequence in a DNA. So when we run that terminated sequences under electrophoresis condition, because DNA, based on its molecular weight, we can separate. So we will be having a ladder of DNA with a terminated sequence like T over here, G over here, C over here. So we'll be able to identify based on the kind of fluorescence which comes from the particular band of nucleotide. And we can say that, yes, this is what the sequence is there in this particular genome is. And this is really time-consuming method, but it is gold standard and traditional method. Even people do this kind of sequencing nowadays within the lab also, because it is easy to do within the lab. But when we use sophisticated and cost very expensive system, we have to go for second generation of sequencer or third generation of sequencers. So when we talk about sequencing, it is simply determining exact order of nucleotide in a given DNA or RNA or gene product. Even we can measure from RNA to DNA also. It depends on our methodology, how we are going to sequence a particular genome. So next-gen sequencing platform can perform massively parallel sequencing in millions of fragments of DNA. Whereas Sanger sequencing, it is mostly applicable for 1 or 2 strands. But yes, in case of second and third generation sequencer, we can use multiple copy or different copy number or millions of DNA copy number and then do the sequencing in very fast way. So it is all about the irreversible chain termination method by dideoxy nucleotide sequencing in case of Sanger. But when we have multiple type of copies of DNA, we can go for NGS. Simply, it is one can easily say that yes, 1 read per sample in case of Sanger sequencing is easy. But when we want to go for high throughput, yes, we have to go for next gen sequencing. And mostly in case of cancer, we go for targeted sequencing because we know what kind of element is there. But in case of targeted sequencing, we will not be able to identify if any specific kind of change is there in transformed cells or not. So whole genome sequencing are chosen when we are not able to identify that kind of a particular type of mutation or change in genomic material after running targeted sequencing. So simply, if we talk about the basic, we can divide NGS in 4 parts. First thing is we have to collect the sample, isolate or take out the nucleic acid, either it is in the form of DNA or RNA, then prepare a library. Library, simply, it is all fragmented DNA that is isolated from the fragmented nucleic acid that is taken out or isolated from the sample. We'll discuss all these steps one by one. After taking out or after preparing library, we need to amplify it by using particular methodology or a specific methodology. We can go for emulsion PCR, we can go for bridge amplification, we can go for rolling circle amplification. Once we amplify and increase the copy number of these genomic materials, we can take them for the sequencing. The sequencing can be divided in various parts, or various methodologies are there, like synthesis or ligation or pyrosequencing or semiconductor sequencing. It depends on us what kind of methodologies we are supposed to select. The most difficult and complicated is data analysis. We have to have very good data analysis part, as well as interpretation, that how to correlate it with particular type of element. So the challenging part is data analysis along with the selection. So selection, it is all up to us that how to go and where to go. So when we talk about nucleic acid extraction, collect the sample, homogenize it. Once we prepare homogenate, we need to isolate DNA. And if our target is DNA, we need to remove RNA from that. So all these things, all these steps are supposed to be taken care during the nucleic acid extraction. So it depends on us whether we want to go for exome sequencing or we want to go for complete genome sequencing, or we have to select for target sequencing. But we have to have good quality of sample. Once we have sample isolated or eluted from the patient material or the material, we can take it for the library preparation. So now when we consider about the library, we have to go for either it is non-targeted library. Suppose if we need to identify new trait or new kind of mutation, then we may want to go for whole genome sequencing or isolate RNA sequencing sequencing from isolated RNA. And if we know that, okay, this particular mutation might be there in a particular type of cancer, then we can go for targeted sequencing also. So various panels are available for targeted sequencing. And we can go for exome sequencing also. It depends on us what kind of sequencing we are supposed to choose. But everything should be looked into in favor of patient. Yes, how economically it is going to be, or very much supportive for the patient also. So when we talk about isolation of DNA or sample, DNA or RNA from the sample, we collect it, we prepare the fragmented DNA or DNA fragments from that. Once we have fragments, we need to tag them with a particular adapter molecule. Once we tag it, we need to amplify it. And amplification is simply like PCR. So we have double-stranded DNA, and all these tags will be related to or will designate a particular type of sequence in a genomic material. So these tags will be taken, will be used for clonal expression and other expression in next step. So first step is isolation of genomic material, then prepare fragment. And after preparing those fragments, you need to tag them. Once you tag them, just you need to go for clonal amplification or other method that you need to choose. So when we talk about DNA fragment or flank fragment, suppose this particular fragment is there and another fragment is this one. So we have different type of fragment. We need to simply amplify them. Amplification, just now we have seen over here, it is all about the PCR. So everything is done at a particular temperature. So it is supposed to be done in isothermal condition. So we need to provide a solid surface coated with a particular complementary primers or primer sequence, which is complementary to the adapter or linker proteins which were tagged in previous step. After that, isothermal amplification is there. In that same temperature is used for amplification of all these primers. So it is simply like a primer binding with a complementary sequence, and then its amplification. So all these things are done. So we'll be having group of genomic material for the of similar kind of fragmented genomic material, and these can be taken for the sequencing. Another one is bead-based sequencing or bead-based amplification. In that also the fragmented or complementary sequence, those tagged sequence which were available, we are supposed to incubate them on a particular bead. So each bead will be having similar type of fragmented DNA, and all these beads can be separated and then taken for the sequencing. So first step is isolation of DNA, then prepare the fragments or genomic material is isolated, then prepare the fragments and then take them. The third one is either go for clonal amplification by using emulsion PCR or by using bridge PCR. So this amplification is there under isothermal condition. It is supposed to be done. Once we have all these beads or solid surface with the amplified products, we can take them for the sequencing. So now next step is sequencing. All this sequencing, there are multiple methods, but I'm going to talk about the sequencing by synthesis and pyrosequencing. Pyro means release of pyrophosphate. And the energy can be quenched or fluorescence can be measured after release of pyrophosphate and incorporation of a particular type of nucleic acid, nucleotide. So when we talk about nucleotide, it is there are 3 phosphate available on each nucleotide. And as soon as these 3 phosphate, 2 phosphates are cleaved from the nucleotide, you will be having energy released from the nucleotide sequence as soon as it gets incorporated, and you can easily, or one can easily measure the fluorescence as well as type of nucleotide which gets incorporated in the DNA or your extend DNA when you sequence it. And similarly, in case of fluorescence, you have different type of nucleotide. Each is tagged with the specific type of fluorophore. As soon as they get inserted, this fluorophore emission can be measured simultaneously. Once emission is measured, these 2 parts, one is the extended part, another one is fluorochrome, they get cleaved by the enzymatic method, and then the amplification can be seen easily or measured easily by the sequencer. So there are different platforms like Illumina. They offers a specific platform. In case of like we have discussed that we have genomic material, we have fragments, and these fragments are tagged with the specific sequence of nucleotides, and then it is available on a solid surface, and we can easily sequence them. So there are different methods available at the same time. When we talk about ion torrent method, now we know that small change in pH, if any pH change is there, we can easily measure when we have a specific pH measurement probe. So incorporation of nucleotide while its extension or while synthesis of DNA, when we want to measure the pH, change in pH, as soon as the nucleotide gets incorporated or corresponding nucleotide gets incorporated in the genomic sequence, while amplification, the change, as soon as you measure, you will be able to measure a particular type of nucleotide insertion. Now, in these steps, if you look on this particular method, insertion or like filling of same kind of nucleotide like adenine or guanosine or cytosine or thymidine. So these needs to be flushed out one by one. Okay, if one gets incorporated, it can be measured in terms of change in pH. And if it is not incorporated, there will not be any change in pH. So this movement or flush can be measured by the specific pH probe, and then we can identify a particular type of sequence within the genomic material. So these are the methodologies which are equally important during the sequencing. The 3rd generation methodology is completely different and very, it doesn't require, what to say, the number of copies that we amplify and other things. It can easily read from a double helical structure, either it is through a nanopore method or DNA polymerase methodology. So NGS application, when we talk about genomics, in case of DNA sequence, we can easily measure the change in mutation or any kind of change in nucleotide, single nucleotide polymorphism or replacement of nucleotide or indels formation or copy number or translocation of genomic material within the chromosome or extra chromosomal zone may be identified. At the same time, if any changes are there in case of RNA, we can go for that one. chromatin immunoprecipitation assay or methylation of DNA, which are responsible for binding or epigenetic changes, can be identified with the NGS applications. So what we have discussed so far, what exactly NGS is, NGS workflow, the nucleic acid extraction. So what actually is important, we should know what kind of material we are supposed to take, where from we are supposed to take. If you have material, how to extract DNA or RNA as per the requirement. As a clinician, we should know that, okay, this much material will be sufficient for extraction or not. And quality of material is equally important. If we are able to get these materials, how and what kind of methodology or procedures the vendor or person is going to perform in pathology, that how to extract DNA or RNA. And if we are going to work with the DNA, we need to remove or eliminate completely RNA part from the sample. Or we can keep both the things for emergency use if we don't have much sample. At the same time, we need to keep library for the futuristic use. So if we want to have library, we need to amplify it by tagging it, and then use a specific platform. In case of cancer, we usually go for target-based sequencing. But if we continuously do that thing, then we won't be able to identify new targets and new kind of sequence or new kind of changes in a particular type of cancer. So we need to consider good quality material. At the same time, we need to look for specific methodologies for the cancer selection also. And thank you so much for giving me opportunity. Jai Hind, Jai Bharat.
So thank you, Dr. Damodar. We have a few questions in the chat box, but because of time, I think we should take it at the end in the panel discussion. Okay. And as Dr. Toprani sir has left, as he was busy in some other meeting, so I take this opportunity to invite our next speaker, Dr. Gohar. Siddhan, can you please play the slide? Yeah. Okay, so for the next thing, we all know that these, novel high throughput technologies, they generate an extraordinary, I would say, a humongous amount of, data, all of which may not be very important or very relevant to us. So it's very important for us to know how do we interpret and understand the result. And for this, we have Dr. Gohar Shafi from Mumbai. He is the Chief Informatics Officer from One Cell Diagnostic. So Dr. Gohar, please.
Thank you for giving me this opportunity. So as you know, we were discussing about the next generation sequencing and the amount of data we generate through next generation sequencing. The most difficult question is how we'll interpret and how we'll use it in a daily practice. So this is the typical workflow of NGS. When we think about NGS, whether it's an whole genome, whole exome, or it is a targeted NGS. There are two critical parts in a whole workflow which I must here highlight. One is that what we want to sequence. Is it one gene? Is it 10 genes, 100 genes, or whole exome or whole genome? That is very important. And in oncology, we are not going for whole genome or whole exome as of now in clinical practice. What we do is we go for a targeted selection, and therefore this becomes very important. What are the genes, how these genes have been selected? Are you selecting only exonic version, as previous speaker was talking about, are we selecting introns? So what difference that make, if I have only introns and exons, or if I have only exons, or if I have some additional markers? And then our previous speaker has spoken about the extraction, library preparation, and sequencing, and then comes the analysis part. Analysis is categorized into three parts, which is primary analysis, secondary, and tertiary analysis. Primary analysis and part of secondary analysis has been it's well established industry standard are there, how you do when you multiply number of samples together, how you demultiplex, separate them accurately without mixing up them. then calling those variants, doing the quality check, aligning with the then with reference genome, and identifying the variant calling, what we call an annotations. And the third, which becomes most important in daily oncology practice, is tertiary analysis, where you prioritize your variant, classify your variant, and then use an interpretation. And what I call is, there are two types of interpretation. One is analytical interpretation, other is clinical interpretation. So this analytical interpretation, it happens when some bioinformatician is looking into the data and does the analysis, use various rules to classify, to annotate these variants. And then the clinical interpretation happens in the mind, in the head of an oncologist who is going to treat his patient. So these are the two critical interpretations, and what I believe that these two should be integrated. There should be a solution that where, and after doing analytical interpretation and generating a report, an oncologist should have the ability to see why this mutation has been incorporated into this report. What is the reason behind that? All this. And if time permits, I'll show you some of the application which we have been developed, which can actually change the way the NGS practice is happening in oncology. The problem is, we know that one size doesn't fit all. We know some of the patients respond very well to the same therapy, having the same disease, same stage. Some don't, and some show adverse events. So how do we address that? So how to improve prognosis, and how to identify patients who need a treatment, which can actually, where we can avoid the toxicity. So what I call is who to treat. We should know who to treat and how to treat. And second is how to identify the effective treatment opportunities, which are tailored to an individual patient. So that's how to treat. So these are the two big questions, who to treat and how to treat. And this is where actually NGS is playing an important role. And we have seen tremendous improvement in last decade that NGS has made in terms of treating cancer patients. So this is all because of the variation we see in the genome. But the issue is, there is a complexity of these variants and how we are classifying them, and there is a heterogeneity, and these challenges create as how should we use this information? What we know earlier, we were calling, there's a mutation and there is a polymorphism. The mutation means it's a disease, is a clinically relevant, it's a disease causing mutation. Polymorphism means which is not clinically relevant, and which is often present in most of the populations. But the new theory is completely changing based on the clinical question. There may be a family history of patients. There may be patients who where we found a mutation, and that could help us to identify minimal residual disease. There may be, the mutation can be a germline mutation, can be a somatic. All these number of questions arise, and now then it becomes difficult for an oncologist to, how should I read this kind of information and how can I use that information? That's why we do.
I think, sir,
I think
he is stopped. Just a minute, let me
Just check.
so. In the meantime, I think Dr. Manjulata, is it RPCR or RT-PCR? Just there is one question in the.
Yeah, I suppose she might be asking about the role of RT-PCR in the NGS.
Okay, so is she available? I think RT-PCR is very much important. When we talk about RNA or exome sequencing, then we do product in the form of RNA, we need to convert it into the DNA or cDNA. From that only we can amplify and prepare number of copies of similar gene product or similar copies of a particular gene product, and then we'll be able to tell that what kind of sequencing it has. Yeah, Dr. Gohar is
Yeah,
there.
Dr. Gohar has joined.
Please take on. Kiri.
Yeah, so sorry, there was some network issue. So what I was talking about, in 2003 we had a lung cancer as a one disease, and in 2022 we have lung cancer with at least 13 genomic alterations which are as per the NCCN guideline. That means we have now lung cancer as a 13 diseases. So these based on this genomic alteration, we have to treat the same lung cancer disease which was in 2003 as one, and now 13 different ways. So what are those genomic alterations? If I summarize that, it could be a single nucleotide variants like we know BRAF V600, or it could be an insertion or deletion like it's a 19 deletion what you daily use in practice. It could be an amplification or expression like HER2 amplification. It could be a fusion like ALK-EML4 fusion or ROS1 fusion. It could be tumor mutation burden. New markers have come up, which is important. Immunotherapy biomarkers, microsatellite instability, and now we also know, loss of heterozygosity, homologous recombinant deficiency, all have become important. Now, how we can analyze, interpret these reports. Now, this is a theoretical representation of when we look at the data, while we do the analysis. So what you see is here, like this at the top is the reference genome, and if it was supposed to be an A, and then in the sequencing, when we do it, we see the change to the C. It's called point mutation. And if this, which is missing here, it calls the indel. And if this whole portion is missing there, it's a homologous deletion or a loss. And if it's partially left, it's a hemizygous deletion. And if it is more sequence, it's a gain or amplification. And if two chromosomes or two parts of the genes have come together, it's called translocation or fusion. So now how we can categorize them? We need evidence, and so that we can actually do the interpretation of these variants, these genomic alterations. So interpretation of every variant should be focused on their impact on the clinical care. That's very important. And how we do it, what are the steps for that? So we should have a sufficient amount of evidence of that variant having clinical significance. Then we can determine whether the impact of that variant is a diagnostic, prognostic, therapeutic, preventive, or a resistant mutation. And we need to evaluate the strength of the evidence for the clinical impact. So how we do it, there are two parts. One is that we look at the variant side, the other we look at the clinical impact. And variant side, I first need to understand what is the type of the mutation, whether it's an activating mutation, whether it's a loss of function mutation, is it missense mutation, is it nonsense mutation, is it indel, is it splicing variant, is it copy number variation or amplification, fusion. So all these, when I understand that, then my second question is it present or absent in somatic database like Cosmic? If it is present there, what is the variant allelic fraction? When we do the sequencing, how many reads support that variant? And if is it a potential germline variant? Like, if I have a variant allelic fraction 50%, close to 50%, or close to 100%, and then I will go back to other databases like germline database, 1000 genome 80 database, see the percentage of these mutations there. Is minor allelic frequency less than 1%, greater than 1%? Again, there is a debate over it whether we should use 1% as a cutoff, but I'll show you that again how we utilize it and see whether present or absent in a population databases. Then functional characterization, how it has been studied, how many publications are supporting there, what is the population it was studied, whether it was studied in Western population, Indian population, different populations, how it has been studied. And then we have some computational prediction tools which predict the damaging or pathogenicity of those variants. Has that been done? And lastly, but very important, which pathway it is involved? Is it the kinase pathway? Is it the cell cycle pathway involved? So all these we try to understand completely, and then say, oh, this variant is very important. And then on top of that, we apply ACMG rules, which classify this variant into 5 categories. Pathogenic, likely pathogenic, variant of unknown clinical significance, likely benign, and benign. And once we have that information, now we see if there is a any therapy which is approved by guidelines like FDA approved therapies, professional guidelines, NCCN guidelines, ESMO, ESCO guidelines, and then how many studies are powered, which support that, that variant and the drug association. Is it investigation therapy? Like, is there any clinical trial going on? And what are the clinical trials? Is it in that particular tumor type you are studying? Is there any small studies, which can show? Is there any case report or preclinical studies? So that gives us, sufficient information. Then we can define whether it's a diagnostic, prognostic, predictive, or theranostic and preventive biomarker. Then we have to evaluate and find the strength of clinical or experimental evidence for these variants. We have as now well-defined criteria level A, B, C, and D. So level A is typically FDA and NCCN guideline approved or professional guidelines like ESMO, ESCO approved. Level B is when we have a good amount of studies supporting that variants with a huge cohort size. And level C are small studies, maybe some clinical trials, and level D is the preclinical studies or some case reports. And how we convert this variant information, what we have seen, into these here. So, and we have a classification of tier classification, tier 1, 2, 3, and level A and B comes, in the tier 1, where we have the variant is a strong clinical significance and is an important and should be acted on. Whereas tier 2 is C and D, where variant is of potential clinical significance. And tier 3 is unknown clinical significance, which may change and has to be carefully looked into based on the clinical presentation of the case. And tier 4, which should not be reported at all because they are of no use. And how we interpret, it's very important. When we do the interpretation, I had to have a correct pathological diagnosis. What site the sample has been taken, and subtype, stage, grade. It's very, very important. And most importantly, intent of use. Particularly, when you have carcinoma of unknown primary region, is an oncologist looking for information which can help him to guide the treatment, or is it looking, why this patient is not responding and progressing on that, identifying the mutation of resistance. And then what is the evidence I have? What is the function that mutation has, gene has, how it is involved, variant, as I spoke about, mutation type, MEF, all that, clinical significance. And then there could be a co-occurring mutation. We have now seen like EGFR, L858, or exon 19 deletion, or co-occurring with TCR90M and showing resistance to first and second generation inhibitors. So that becomes very important, how my co-occurring mutation may lead resistance to the main target therapy, which is based on the driver mutations. So, and then, how we have additional informations. And how we deal with the polymorphism. Earlier we were talking polymorphism are not important, greater than 1%. Again, it becomes important. Here is one of the example, which is very important. Here if we see this KDR-Q72H is an polymorphism, and the population frequency of is in exact 22%, means 22% of people, healthy population have this mutation. But there are reports where they have seen that anti-angiogenic treatment in melanoma patients have done very well on those patients. So therefore, such information has to be carefully looked into, while doing the interpretation. And, we have to follow the guidelines, though we are talking about, we should have matched samples, but that's not often possible. Therefore, we have to be very careful when we have a mutation, which may be in genes, which may have a germline potential. And so that information should be communicated and explained very well. So, different labs have different policies, but it's very important how you are disclosing information, particularly germline variants, how you are communicating to the patient, and how you are counseling their patients, how that should be utilized, in order to make sure that, they get appropriate counseling. In silico, as I was talking about, in silico prediction is also important when you have the variants to identify, you may not have sufficient evidence, but there is an in silico prediction showing the potential of that variant has a damaging effect. How we can utilize, particularly, it's very useful when we have variant of unknown clinical significance. And Yeah, I think I just want to, I know this time, so just one or two examples. This is one of the example, high-grade salivary gland ductal carcinoma. And, if you look at this patient has amplification EGFR gene, almost more than 70 copies. And this patient, when it was treated with the EGFR inhibitors, has done very well. That's how this becomes important. Another example here is non-small cell lung cancer, and the mutation found in the MET gene, though not the classical exon 14 mutation. So what this mutation was found in, it was 12 base pair deletion in MET intron 13. That's how introns become very important, and hardly there was any literature, not well characterized splice site variant like exon 14, and VF variant allele fraction was 20%, but minor allele frequency of this variant in population database was less than 1%, and MET intron 13 deletion was overlapping actually region that has been studied, particularly like MET exon 14 skipping, and where you have seen the crizotinib like MET inhibitors to have done well. So this was classified and variant of unknown clinical significance. So this becomes very important, how you can utilize unknown clinical significance variants, and this patient actually could benefit with MET inhibitors. So with this, I would like to thank organizers for giving me the opportunity, and if there are any questions, I will be happy to answer those questions.
Okay. So thank you, Dr. Gauhar. I must say that was an excellent speech, and we now understand, I think, something. How do we interpret the enormous, information we get from those genomic sequencing? I think we have a few questions. There is one question from Dr. Rutesh. Sir, according to you, which method is more reliable for sequencing to study the mutations in DNA?
It depends on our pocket. It depends on your budget, number one, and what kind of sample you have. But yes, you can go for any sequencing which gives you best possible result as per requirement, because all methods which are available in the market, they are there because of their importance. Say the clinical importance as well as sequencing also. It depends on us that which particular method we are supposed to select.
Okay.
Whether you go for targeted, if you knows about the target, or you have to go for whole genome sequencing or particular methodologies, depends on exome or other DNA targets. Yeah, depending on type of mutation also it is equally important. And what is the length of a target sequence, that is also important.
Okay, so thank you, Dr. Damodar and Dr. Gauhar. Please stay connected. Now it's time to go to the second session, and I take pleasure in inviting our chairperson, Padam Shri Dr. J.K. Singh sir, a very well-known and famous oncologist. Sir needs no introduction, and to co-chair the session, I invite and welcome Dr. Sanjeev Mishra sir from Lucknow. So please.
Very good evening to all of you. And I think we'll have 20 minutes time, and we'll have a very important topics to be discussing regarding the tyrosine kinase inhibitor pathway. And friends, if we talk about the tyrosine kinase inhibitor, it blocks the chemical messenger and also the enzyme called tyrosine kinase, which signals the process within the cells and blocks the process of cell growth and division. Tyrosine kinase is a mediator of signal transduction process, which leads to cell proliferation, differentiation, migration, metabolism, and programmed cell death. I think the different issues related to tyrosine kinase pathway, we have a very eminent speaker. He doesn't need any introduction. This is no other than Dr. Dinesh Pendarkar from Delhi. I think he will discuss and he will enlighten us about the different steps of the tyrosine kinase pathway and its mechanism of action also. So I would also like to, my co-speaker, he will also like to tell something about this. And then I would invite Dinesh Pendarkar. Dr. Sanjeev, please.
Thank you, Dr. Singh. I think we are running late, and I think Dr. Pindarkar is the best person to talk about tyrosine kinase pathways, and he's a very eloquent speaker. And over to I would request Dr. Pindarkar to give his talk and enlighten all of us on this issue. Dr. Pindarkar, sir, please.
Thank you very much. Am I audible? Can you share my screen?
Yes, sir. Yes, sir. You're audible, sir.
Audible.
Can you put my slides?
Should I share a slide, sir?
You can put my slides, please.
Okay, sir. Just a minute.
Yes, please. So the most important thing today, what is happening in oncology is we've just heard from everybody that the most important thing which happens in a cancer is a mutation, which produces some proteins which indirectly produce the growth. So naturally, we have to think of mechanisms, how to block the pathways, how to stop this growth. And the best way to do this would be first to understand how the growth actually is controlled. And the biggest pathway which controls this mechanism of growth and cell proliferation is tyrosine kinase pathway. Next. Can you put my next slide, please? Hello.
Yes, sir. It is.
Hello. I don't see that.
Just a minute.
I'm not seeing the slide. Can you see the slide? I don't see the slide.
It's there now, sir.
Is there? Unfortunately I'm not able to
It's
see.
related to clonal origin. It may come.
Yeah. So okay, I'm just doing it. So basically what we should understand that all cancers are of a clonal origin. So basically a disease starts in a one cell, one clone, and then it grows. That's the most important information. But yes, as it grows, it may acquire more clones. So nearly all cancers start in a single cell. That's very important. And mutational changes are needed for transformation from a normal to a cancer cell. So unless you have a mutation, the growth will not be triggered. Next slide. So what are these signaling kinases? So what happens is even for a normal growth, normal cell to continue living, it requires production of some proteins. And this is all done by signaling. So there has to be some signal in the extracellular space which will trigger a cell to do some action. So generally, when we are talking of oncology, we are talking of a malignant transformation or of a cell converting to a cancer cell. Generally, these are tyrosine kinases. All the receptors, the cells have got receptors which have a base of tyrosine. So these are tyrosine kinase receptors. And every receptor has got three parts. I'll show you the slide later. So it has a part which is above the cell membrane called extracellular. There is a part which is inside the cell membrane called intracell. So it's transmembrane. And there is something inside the cell called cytoplasm. Next slide. How these changes happen? The changes happen because of mitogens. So there are mitogens which can actually trigger cancer. Next slide. And But all of us remember the biology we have learned that everything for a kinase, for a protein to become active or inactive, it involves phosphate molecule. So once a phosphate is attached or detached, the molecule becomes active or inactive. That's the basic fundamental principle of a cell mechanism or a cell movement, I would say. So what is required for a cell to grow is the moment it gets a signal, for example, signal for growth, it has to start somehow using this phosphate mechanism. So that's what a cell does. So tyrosine kinase mechanisms are basically a mechanism by which a movement of a phosphate happens from one place to other place. Next slide. And these functions of tyrosine kinase that we can define them, what tyrosine kinase do in the body, they actually can play a role of growth factor signaling. And basically, we are interested in oncogenesis. They may actually produce signals for cell adhesion, whether a cell should stay together with the other molecule or not, whether a cell can move or not, migration, the differentiation of the cell, all these signals start from a receptor called tyrosine kinase receptor. And the whole cell cycle is controlled through this mechanism. So, as I said, for a change to happen in a cell, you need a protein which is moving the cell. And the protein is there, but it is inactive. A protein becomes active only if a phosphate molecule is attached to it. Next slide. Next slide. So, what are the roles of these tyrosine kinases? Gene regulation and transcription, endocytosis, something like angiogenesis. All these mechanisms which are required for a cell to survive and grow are controlled by this simple mechanism of phosphate change. Next. As I said, there are multiple signal transduction mechanisms. It could be on a cell surface. Yes, please. Hello.
Yes,
We
sir.
can
You are
hear you, sir.
audible.
I can't see the slide again. Gone.
It is.
Doesn't matter. Okay. Okay. So don't worry. So, so what happens is these receptor tyrosine kinases, that's why they are known as receptor tyrosine kinases, they transduce growth factor through the cell and they regulate the gene function and they trigger cell proliferation. So what is important for us as an oncologist is the knowledge of these tyrosine kinases. Mentioning two because they are known to you, but actually there are multiple tyrosine kinases available and they have got three portions: extracellular like transmembrane portion and a cytoplasmic portion. So the structure is three things. So the binding of the growth factor receptor, whenever there is a growth factor, it binds to the receptor. So it will bind to the receptor. Next slide, please. Next slide. Next slide. And the next slide. So this is how the structure looks like. So this red one is a growth factor. Connection with a receptor, generally and two receptors have to come together to start.
Sir, sorry to interrupt you. You have a weak internet connection. Can you switch off your video?
Together in the extracellular domain, and the signal will go inside. Here at the end, there is a tyrosine molecule, and it activates the association of a phosphate. So the phosphate is exchanged. Next.
Sir has left. due to poor internet connection.
Hello. So what happens is whenever this phosphate is available here, then there is a special molecule called GRB2 which absorbs, which gets this additional phosphate. So GRB2 otherwise is a non-active molecule. The moment it gets an additional phosphate, it connects with a molecule called SOS. This process remains same everywhere in the complete cell. And once this is done, there is another tyrosine kinase called RAS kinase. So RAS kinase is a basic molecule of majority of the processes in oncology. As I said, so as you can read here, it's a RAS GDP. So it's a diphosphate. So once an SOS molecule brings a phosphate to diphosphate, it converts to a triphosphate. So the moment proteins become a triphosphate, it becomes an active protein. So now imagine if there is some mutation and this process is not happening, that means there will be a constant availability of triphosphate. So if there is a constant availability of triphosphate for whatever reason, either there is a growth factor constantly acting or there is a mutation in the RAS gene, naturally everything will start growing and there will be a proliferation of the cell. Next slide. So this autophosphorylation which happens after dimerization is the initial. Next slide. Next slide. So this in a short, I'm just showing one pathway, but the first important thing is dimerization, then an activation of RAS pathway. The moment RAS becomes active, it makes another molecule called RAF molecule active, then it makes MEK molecule active, then it makes MAP kinase active, and the MAP kinase is a capacity to enter the nucleus and work with the genes.
So you're not audible.
Sir has very poor internet connection, so it's got freeze. Sir left again.
Okay.
So once he will come, we'll request sir to switch off your video due to the internet connection. The bandwidth is low. So let the video have switch and we.
So in the meantime, can we take a few questions till sir connect? So I think we have a question from Rupak Kumar. Please address the difference between primer and probe used in cancer molecular biology. Is it commercially available or customized? Dr. Gohar or Dr. Damodar, can you please take the question?
Yeah, so there are, when Dr. Damodar was explaining various sequencing technology, and there are steps involved. If I take an example of hybridization method by Illumina, and there is a first step, where I had to do amplification and primers, random primers are used to amplify entire DNA. And then if second step, if I use targeted sequencing where I have a selected number of genes, I had to have a probes to enrich only those regions to have now, library prepared for only those regions. So probes are used to enrich the specific region of the DNA, and primers are used for amplifying entire DNA. Now, the second part of the question is, is it possible to customize? Yes, this is possible to customize. This is what my first slide was, target selection. If you want to design, for only 1 or 2 genes or 5 genes, you can customize, and you can design what regions you want to actually do the sequencing.
So we have another question. Kindly mention if there is a source or a book where I can learn data analysis of NGS in detail.
Yeah, very good question. Yeah, there are a lot of resources available. See, data analysis, as I said, there are 3 components of data analysis: primary analysis, secondary analysis, and tertiary analysis. So primary and secondary analysis, a person must have a coding knowledge, like Python, R, and Perl coding knowledge should have. And there are various—
Dr. Pendarkar sir is here.
Yeah, I think sir is.
Yes, Dr. Dinesh.
Yeah, it's fine. So I think, I think I'll go to the last slide and this slide. And I would just like to tell you, we should remember that this signaling pathway is a big pathway, long pathway, and there could be mutation at any place. And when we want to intervene, we can intervene at many places. So sometimes when the drug doesn't work, it happens because if you are acting at a RAS level, the RAF level starts working extra, and there is no role of your drug. So there is something happening because of you. So the downgrading pathway becomes more active. So we should remember that all these tyrosine kinase pathways, like in biology, everything works with a feedback mechanism. The moment you block somewhere, the extra thing goes. So I think I would like to stop here because really bad connection. But I think that's all I would like to say. Thank you.
So thank you, sir. I'll request the chairpersons to please have a comment and then start a case discussion.
Thank you, sir, for giving us a wonderful overview about the tyrosine kinase and how they work and how they act. And I think it's important to know the mechanism and how you develop the resistance to them, because if you know the pathways, then you know how the cancer cells will bypass them. And thank you, Dr. Richa and Dr. Damodar, for asking me to chair the session, and Dr. Singh, and back to Dr. Singh and Dr. Richa.
Thank you, sir, for taking time out. Thank you. So before, I think we asked Dr. Neeraj Arora to start the case discussion. Can Dr. Gohar, can you please complete the answer? Where do we learn the NGS?
Yeah, so there are, now the thing is what a person has to learn, that's what, primary and secondary analysis, coding knowledge is must. And if it is just, a tertiary analysis, and there are various resources available, and, they can get in touch with me and I can provide them resource links where they can read about it and understand how we can do the interpretation of as well as tertiary analysis.
Thank you, sir. I think there are a few more questions, but we'll try to take them up in the discussion. So now I invite Dr. Neeraj Arora, he's a director of molecular genetics, to start the case-based discussion and invite his panelist. Dr. Neeraj?
Are my slides visible?
Yeah, we can see the slides.
Yeah.
Okay.
So at the outset, I would like to thank the organizers, Dr. Richa, Dr. Damodar, and ISO for giving me this opportunity. What I've done is I have kept it very basic because for the this is mainly for the students. So if you find very basic, so please you can skip because this is mainly with respect to some of the oncology students is what I have kept it in mind. And I would like to welcome the panelists, Dr. Dinesh. We have already heard, sir, putting across the kinase domain in that. Dr. Arvind from Chennai, Dr. Arvind Krishnamurthy from Chennai, Dr. Sumant Gupta from Delhi, Dr. Gohar Shafi, we have already heard, and Dr. Damodar Gupta from Delhi once again. I would like to welcome all the panelists and would like to see if they're all there.
Dr. Arvind has not yet joined, and Dr. Suman, both of them, they're expected to join soon. So I think you can continue. Thank you.
So we have.
I'm around.
Yes, sir. Yeah. Yes. So we have Dr. Damodar, Dr. Dinesh, and Dr. Shafi with us. Hopefully, my panelists are going to join soon. And we will straight away go to the cases. These are very basic cases. So case number 1. So this was a 50-year-old female who had a biopsy done, and on the biopsy from the intra-abdominal mass in the abdomen, this is the markers that we got, that is CD117 patchy positivity and DOG1 strong diffuse positivity. It was a spindle cell tumor, and the Ki for this cases was around 3 to 7%, and they were negative for S100 pan-CK CD34, and it was labeled as low-grade gastrointestinal tumor. So it was a low-grade just in a 50-year-old female. And so the questions for the panelists, I think first question is, so if you have got a GIST, will you do any additional molecular tests? Do you do it, or anything you would suggest to do it? Dr. Dinesh, if you are there, can you please take up that question? I think Dr. Dinesh is not there. Dr. Damodar, will you like to comment on that? Any tests?
Course, one should go for sequencing again and look for the entrance part also.
Okay.
Along with the exam.
So any specific genes in GIST implicated where which has got any implications for therapy? Any specific genes you would like to do?
To me, one should look for a particular kind of expression in the cell. First, the physiology needs to be identified because it is completely heterogeneous mass of a cell. So what is going to happen at the cellular level or subcellular level, that needs to be looked into. So it may be epigenetic changes also. There are chances. So
Dr. Shafi, would you like to take up that question? Any molecular tests you think?
Yeah, definitely. I feel, we should go for NGS test, preferably, comprehensive one, which has genes covering mismatched repair and which can capture microsatellite instability. And I would go a comprehensive approach here.
So traditionally, usually in a GIST case, I mean, there are two genes which have been implicated. One is the c-KIT and other is the PDGFR. Two of the commonest genes which have been implicated in gastrointestinal stromal tumors, and the GIST, the c-KIT gene which is implicated is predominantly done with respect to the therapy. So if there are mutations, some of the mutations, they do not respond to therapy very well. So that is why these two genes have been kind of done or recommended when you are doing a GIST. And as you had already mentioned, Dr. Shafi, that now we are moving from a single gene testing to a more comprehensive or a larger panel NGS based testing approach. I agree with you that in these situations, single genes testing by a Sanger sequencing may not be able to pick up some of the variants at which are at a variant allele fraction of less than 20%. So you are likely to miss out some of them, and you can have negative cases when you are doing that. So Dr. Shafi, if I would like to ask, as you already mentioned previously as well, what are the common approaches for sequencing using next generation sequencing?
Yeah, so in oncology we go targeted sequencing, and there are two approaches. One is hybridization method like Illumina we use, and the other is amplicon based method like ion torrent thermo panels. So we have to consider if I'm looking at just SNVs and indels, point mutation and insertion deletions, then amplicon based approach is good. And if it is in DNA RNA panel combined, then it may give us fusion information as well. But if we want to have a DNA based translocation approach, hybridization approach is the best approach to go with, which is Illumina based platform. That's what I understand difference between the two different approaches currently being used in oncology.
So what Dr. Damodar was mentioning, and Dr. Shafi was also mentioning, with respect to what approaches you can use, you can have a whole genome approach, you can have a whole exome, and the targeted approach we just discussed. Then there is an RNA based approach or a transcriptome based approach. You can use any of them, but whole genome, exome, and transcriptome primarily in the research domain as yet. We use exomes in the rare disease settings, but in terms of oncology, as Dr. Shafi pointed out, you do not use. Mainly, we are more focused of targeted approach because they are very fast, cost effective, and they work on very small amounts of FFP. With respect to two approaches, like it is either an amplicon based or a hybridization based approach. So they work very well with the FFP tissues, meaning once you have tissue, you make blocks and use it for a histopathology testing. And from those FFP blocks, you are able to extract DNA or RNA, and further downstream, you can do all the tests that you do.
I'm sorry to interrupt, but Dr. Arvind has joined. So
yeah. Okay, thank you. Thank you.
Dr. Pendarkar is also available. Pendarkar sir is
sure. That is very
welcome, Dr. Arvind sir.
Welcome, Dr. Arvind. Thank you for being with us.
Pendarkar sir wants to have some comment. I think he has.
Yes, sir.
Yes.
So what I was feeling, definitely when we are talking of whether I do a 1 gene, 2 gene, or a small panel or anything, I think we should start teaching ourselves and everybody that if at all we really want to go deeper into science and oncology and everything, this business of 1 gene, 2 gene, 20 gene panel, 15 gene should go away. There should be a maximum gene panel with whatever is feasible today, like maybe like foundation 350 genes, or it may be somebody else doing 400, 500. I don't mean, but let us start focusing on those panels rather than just doing piecemeal and trying to remember every gene and every disease. I think this is the way we will learn, because it is quite possible that our population might be different. It is quite possible that in our subcontinent, maybe some other mutations are available. And if we continue to do this business of 15 gene lung panel, 20 gene colon panel, we'll actually miss a lot of science. So I think we on our forums should be advocating only and only of maximum gene number or maximum number of gene alterations study possible. Thank you very much.
Thank you, sir. Thank you for your comments. Certainly, that is a very, very forward-looking. If we are able to work in that, I will be very happy.
Yeah, it is very much required even in case of therapeutic response.
Yes.
Also, if you are able to identify new targets, because it is heterogeneous population. That's why I said, yeah,
we look at a more comprehensive level.
Is really difficult for a clinician if any mutation is there elsewhere also.
So traditionally, when we talked earlier, of the targeted approaches, we generally used to mention that only DNA is to be used. But now, with the use of RNA-based targeted testing, we are able to pick up point mutations, insertions, as Dr. Shafi pointed out, and you have also been mentioning copy number alterations and fusions, and even expression. So all of this can be done from a combined DNA and RNA-based testing, and that is what it is happening, is available, easily available, and we tend to do in this. So for the next question, so we are having a case of GIST, and we were discussing, what kind of things we can do. We just mentioned some of the approaches that are there with respect to next generation sequencing. So I have a question for Dr. Arvind. So how do you select a panel, when you're ordering? As Dr. Dr. Dinesh mentioned that you should have a larger and comprehensive panel. So what would actually anyone can answer? I would request Dr. Arvind if he could give his comments.
Yeah, hi. Actually, I'm slightly traveling. I just landed, so I am not really sure whether how my signal would be. But I sort of endorse what Dr. Pendarkar has said. Like now, so going from the now it's the era of precision medicine, and we should try to be as comprehensive as possible. So we have to expand it. Not, rather than having fixed or limited panel.
Thank you, Dr. Arvind. So basically, again, as you mentioned, so there are two approaches. Either you want to have a very focused approach or a very comprehensive approach. Traditionally, based on the profiling or what is information available for that tumors, you would like to select a panel. And with respect to GIST, as I mentioned earlier, there are KIT mutated, there is PDGFR mutated, and there are other genes which have been implicated. And this list is expanding, and we do have NRAS, HRAS, KRAS, and some other fusions being described. So if you are working out a comprehensive approach, it works out very well. But in this particular case, the clinician selected for a smaller gene panel, trying to look at PDGFR alpha, KIT, and BRAF mutations, and they ordered a smaller DNA-based panel, one of the very smaller and cheaper and comprehensive panels available. This is a 50 gene panel, and this panel was done. And primarily, this panel can be used for colon, where we do have KRAS and RAS, BRAF again implicated. You have GIST, as I mentioned, and you also could be used in melanomas where you have c-KIT, RAS mutations being implicated here. You want to say something, sir?
So I want to say, I think we should forget this business of cheap.
Okay.
Because what is happening is, if these small panels are costing you 15, 20, 25,000 rupees. Okay. And a big panel of literally 400, 500 Gs, there are companies which are offering literally at 50,000 rupees. So I see no difference between 25,000 and 50,000 rupees, number one. And if I offer you more numbers, I'm sure you could be able to bring it down even below 50,000. It's already being offered in the market. Number one. Number two, so we should just, because as you just now said in GIST, there are so many other mutations which are possible. So number two, if I find a target here, a minimum target cost starts at 70,000 rupees, if not more. So if I just spend extra 25,000 here, I don't think I'm losing much for fighting for a drug which would be actually likely to cost much, much higher. So I think we have to change our mindset. Thank you very much.
Thank you, sir. Thank you. That is a very excellent statement. So for this particular case, as opposed to what we thought c-KIT and PDGFR alpha, we got a mutation in BRAF. Henceforth, this has got both diagnostic and treatment implications. Just would like to point out that this is V600E mutation, and as Dr. Shafi was mentioning, the coverage, 620x and variant allele fraction 47%. This is a gain of function and pathogenic and tier 1 classification. Just to share with you that those treatment details are mentioned, and you can use that kind of information for the treatment purposes. And when we look at in the laboratory for any particular mutations, we look for the electrophoregrams, and this is how electrophoregram or IGV looks like for a particular mutations, where this is an IGV where we sort of confirm. So any variation which we look up or variant which comes up, we need to confirm whether it is looking good on the IGV, and then subsequently report it in the way it was done earlier. And okay.
Yeah,
so that was with this.
Can I just, in this context, can you take up a question? There is a question that if a sample is likely pathogenic, should we proceed as variants of unknown significance or as pathogenic? Should we treat in the side of caution or wait for more evidence?
See, as Dr. Shafi had already pointed out, that sometimes when it is pathogenic or likely pathogenic, certainly there is evidence to be said that this is within the domain of what is reported. But when you are working in the domain of VUS, means this information is not available, and we need to have more clinical relevant information. As Dr. Shafi had highlighted one case of VUS But with respect to the clinical information, the clinicians went ahead. So that call has to be taken with after discussion and with respect to the clinician. So I think that call after clinical correlation or molecular tumor board meetings, you have to take a call on that.
Okay, thank you.
So this was a second case. So this was 51-year-old female presenting with increased frequency of loose stools, blood in stools, vague abdominal pain, loss of weight, and there was family history of cancer. It was mild pallor, no organomegaly. His hemoglobin was low. CEA was 4.53. Sigmoidoscopy showed a large tumor more than 5 centimeters in the sigmoid colon, causing stenosis. So a surgery was done, that is laparoscopic anterior resection was performed, removing the sigmoid colon or rectum. And this is what we saw in the biopsy. You could see the pools of mucin and the tumor cells over here. And along with it, you could see some lymphocyte infiltrate. So it was a mucinous adenocarcinoma, moderately differentiated adenocarcinoma in the sigmoid colon. So so I would like to ask Dr. Dinesh, is there any role of MSI testing with respect to the colon tumors or colon cancers? Is the role of any MSI testing here?
So my philosophy is very simple. PDL1, MSI, and TMB to be done in every cancer irrespective.
Okay.
Colon included.
Okay.
Because pembrolizumab has been approved for this indication, irrespective of cancers. So I am not bothered about diagnosis of the patient. It's an agnostic treatment, and I want these 3 markers in every patient, and I do it.
Sure. So as already mentioned, that MMR or MSI is with respect to colon is with respect to identification of Lynch syndrome, with respect to prognosis, and it can give information with respect to immunotherapy. And it has been recommended for, it is a tumor agnostic panel, and it has been kind of recommended for all tumors to be done here. And how would you prefer to do MSI, Dr. Damodar? Whether it is immunohistochemistry, PCR, or NGS, or what way would you look at to do MSI testing?
We would go for immunohistochemistry because
and this is all tumors, or in a selected category of tumors you would like to do?
I think for a selected one, not for all.
Okay. So traditionally, I think as was mentioned earlier, for all tumors it can be done because it is a tumor agnostic panel. So even if you don't have any cancers, all cancers can be done. So this is MSS2 IHC, and traditionally when you're looking, the loss is what is you are looking at. And here you can see the normal cells which are positive, but the tumor cells are negative for MSS2. Then the other marker is MSH6, and as you see, the normals are positive, whereas the tumor cells are showing variegated. So this is, I would call, a variegated loss. Not complete loss is there, but sometimes you're not able to take a call. This was variegated is what our pathologist put it as. Traditionally, every cell should be positive, or tumor shall be positive, or it should be retained. But the loss is what is significant. And this is the third marker, that is MLH1. You could see it was retained, normally expressing both the tumor and the normal cells being positive for MLH1. And the fourth marker is PMS6, so it was again positive in the cell. So you had a mucinous adenocarcinoma moderately differentiated with intratumoral lymphocytic response. And on immunohistochemistry or IHC, we had a suspicion loss of MSS2 and MSS6 loss was there. This was suspicion because traditionally when you are calling loss means it is complete loss. It is present or absent. There is no intermediate thing here. What I showed was the variegated appearance, and that was created a confusion. So, and the further testing by a PCR based protocol was advised here. Testing by PCR was done, and this is how you do a PCR based testing, where you need a peripheral blood and the tumor. So both, so you require additional peripheral blood and the tumor DNA to be tested by a panel of markers. Traditionally, this is the 5 markers which are there, and could see on the comparison between the BAT26 on the peripheral blood and the tumor, there are additional peaks seen. So meaning that this particular marker is abnormal, or it is showing some abnormality. The other markers like NR21, BAT25, and MONO27, you need to compare peripheral blood with the tumor, and you can see additional peaks seen over the MONO27. Also in the BAT25, you need to compare peripheral blood and tumor, additional peaks seen over here. Then NR21, additional peaks seen over here. And the 5th marker, that is NR24, you can see there are peak scenes in the tumor tissue. So what do you imply by a PCR based testing is that since this is more than 2 markers is what you consider high. MSI high is on a PCR based testing mode. Out of the 5 markers that I showed, 2 should be abnormal. So in this case, all the 5 markers were abnormal, and the interpretation from this was that so now there is a loss in IHC. It is also showing that yes, there is a loss on the PCR based testing as well. It is also showing that this is high. So can I ask questions? So role of MSI testing in other cancers to Dr. Damodar, or if anyone could take up that question. I think this question I will skip because anybody want?
Yeah, I think it's become very important, as Dr. Pendarkar sir highlighted. This pembrolizumab is approved in all solid indications, and MSI is one of the important biomarker for that. Therefore, it should be done. And nowadays, a lot of NGS panels come with MSI, part of that. So I think we should look into it in other indications as well.
Yes, thank you, Dr. Shafi. I think this is the first tumor agnostic therapy approval is there for pembrolizumab, and we have other markers like NTRK and NRG1s being described in that scene as well, which are tumor agnostic. So if there is a cancer, you can go ahead for those kind of panels and testing. So now you have a mucinous adenocarcinoma, loss is there on IHC, PCR is also there. What do you want to do next? You want to start treatment, or would you like to do anything extra as well? Dr. Shafi, would you like to take up?
Well, I'm not the right person, to decide, treatment. But as we see, the results are indicating, it's an instability is there, and oncologists should go for it based on the results. But it's oncologist call here.
Yeah. So one of the things regarding MSI is that it helps you pick up Lynch syndrome. So since there is family history is there is a lymphocytic infiltrate is there. So proceeding further, NGS for hereditary cancer panel was done. Traditionally, these panels are now available where you can evaluate 100 commonly implicated genes. The sample type is peripheral blood EDTA sample is what you need to evaluate when you're doing hereditary panel exclusions. And what we found here was a pathogenic variant in the ABCAM gene. So just wanted to highlight that there was a ABCAM gene. So apart from the 4 common genes that you see, MLH1, MSH2, MSH6, and PMS2, ABCAM is seen in around 1 to 3% of the cases. And because of the availability of NGS and larger panels available, you are able to get out that kind of information as well. So you need to keep it in mind. There are other genes as well. So I'll skip this and come to the case number 3. And this was a 59-year-old female, and the liver biopsy was done. And the liver biopsy showed a tumor which was compatible with metastatic adenocarcinoma from the gallbladder primary. And there was other possibilities which were considered was this would be a cholangiocarcinoma. So that was what we have got. And as Dr. Dinesh was mentioning, that now we need to go in for larger panels or comprehensive panels, and we are going into that area of comprehensive genomic profiling, where we can access or do whatever information is available. So right from mutations, fusions, copy numbers, genomic loss of LOH, then tumor mutation burden, microsatellite instability, all can be done in one go. And for this particular case, this was done. And when we were evaluating, we got mutation in the collagen A1, then KMT2A, TP53. So these were the pathogenic and the likely pathogenic mutations. But along with that, We also got a copy number variant that is ERBB2. So this is a HER2 new as was mentioned. This was an amplification in that gene, and the copy numbers was 36. So very high copies. And no, traditionally as pathologists, whenever we have IHC available, we tend to do that. When we want to look at that, and we are aware that HER2 new is IHC is available, and we did and immunohistochemistry on this thing, and you can very nicely see 3+ positive cases. So corroborating what we got on NGS, that this is an amplification which is there. So I think I need to ask this question to everyone. We have the views of Dr. Dinesh, that role of comprehensive genomic profiling in the clinics in India. So I think each one of us can take up that. Dr. Shafi, if you can.
Yeah, so comprehensive genomic profiling, it's very important. The reason is most of the cancer cases which are coming to the clinic are at advanced stage, and that small gene panels are not sufficient enough. That's one of the reason, and we should definitely go from the beginning comprehensive genomic profiling, because it can open up a lot of information. However, we have to be very careful that these are very expensive. Every patient is paying out of their pocket, and we have to see how we can use this effectively in routine practice. And second thing is, once we get the data out of this comprehensive profiling, there are certain targeted therapies which are available in India, and if that is found, and then great. And if there are some certain targeted therapies which are not available in India, that becomes another challenge. Either we have to look for some option of getting these targeted therapies on compassionate base, or even the clinical trials options. Those are the challenges are there. But what I have seen in India now, the comprehensive genome profiling is being extensively used in clinical practice, even though we have these challenges.
Okay, Dr. Damodar, will you like to?
Yes, I would like to add few things. See, as a researcher, if I am having a sample, or if I provide a sample, I should know what exactly I want from my sample. At the same time, results should go in favor of patients where from we are getting samples. Number one. Number two, when we want to go for comprehensive genomic, and if patient is not able to afford, based on our company or requirement of that particular patient, even if we are able to identify a particular change or in trait or pathology of that particular patient, or in a sample of that particular patient, it is going to be of immense benefit for future in case of if we identify similar kind of patient in future, and if we have targeted therapy for that person. So yes, going for comprehensive is very good. Going for targeted is equally good. Only thing, it depends on the patient, whether they are able to afford. But yes, as a company person, if we want to look for, we can keep that library in our hand and go for the targeted therapy, because in coming time, like Dr. Arvind said, we have to go for precision therapy. And as a company guy, if you are able to support that particular patient, yes, this kind of mutation is also there, because doctor is not able to tell you every single thing about the patient most of the time in India, because they don't have much time. But if you are able to identify that patient or element in that particular patient, then it is easy to choose what kind of changes will be there in that particular genomic sequence. So yes, going for comprehensive is really good. I agree with that. But yes, it should go in favor of patient. Yeah, Dr. Pendarkar wants to say something. Sir, please.
Yes, sir. Yes, sir. Please, sir.
My only question is, we are going towards comprehensive. We are doing more and more comprehensive. But my humble request to all the labs is, why can't we create an archive where I could visit that archive for my research purposes? It may have no patient name, no patient data, but unless these genomic data goes in one archive, we will still not be able to take anything out of it. So I think all of us should try to work together to get this data in one database.
That is very true, sir. I think, unfortunately, I mean, there are no groups working. If there is a group working, and we can get the data together in one place, and which could be of use to every one of us. Everyone needs that data for a lot of things.
Yeah, one particular center or place can be created through proper channel because it may be related to the individual requirement, also at the same time treatment and safety and security purposes also from that particular sample about their identity and other things. So yes, if we are able to keep a library like comprehensive area or archiving of those sample or keeping those sample in storage condition for futuristic research purposes, it is going to be of immense use for each. But yes, it should be accessible for all clinician and researcher.
So as we are aware that we are moving in that line of comprehensive genomic profiling, and there are a lot of panels available. You can mention TSO, there is Foundation One, there are some customized panels available, there is OCA Plus, a lot of panels are available in that sphere. With respect to Indian scenario, there is certain distinctness that we have to use with care. If it is available, the price is a key factor, and this is one such study I found from India. Clinical benefit of comprehensive genomic profiling, where they did mention that it was done, and around 4% of the patients had a benefit. So very low number of patients, but that is what we are a very diverse country. So even 4% would be Dr. Shafi.
Yeah, I have gone through this study. I feel that this study has used various panels, small gene panels there, and then concluded that only 4% has benefited. So if we are calling comprehensive genome profiling, we should not call a 20 gene panel as a comprehensive or a 50 gene as a comprehensive. It's not given a complete picture. Therefore, it is not appropriate actually to conclude that only 4% have benefited. So if I have done a testing on individuals with 50 gene panel, but that those patients do not have a mutation in those genes, have mutation in other genes. So then I'm missing that information. Second thing is, it's very important, to identify secondary mutations which lead to resistance. So that we miss it in these small gene panels. And I felt that these studies have been poorly designed, and then they have poorly used this comprehensive genome profiling for a 50 gene panel testing.
Thank you for bringing that out, Dr. Shafi.
I totally endorse it.
Thank you. So I think the poster boy with respect to the molecular genetics has been lung cancers. We have already started with lung cancer, and we have come a long way from only EGFR. So if Dr. Shafi could take up molecular, what sort of tests you did mention with respect to the NCCN, lot of tests being there. So could you please describe what tests are recommended for advanced lung cancer where we have gone ahead?
See, NCCN guidelines itself says there are 13 genes now in NCCN guidelines, which they are recommending for a testing. But in NCCN guidelines itself, they have written broad molecular profiling. And when they use broad molecular profiling, means it's not just 12 they are highlighting, but should go for, other than those 12. We have seen patients having, there was a patient 7-8 months ago, lung cancer patients where BRCA1 mutation was identified. Even though the practicing oncologist was not satisfied with the results, but at the end, a patient doesn't want to go for chemotherapy, then he has started olaparib on that patient, and the patient is now on complete remission. So this is showing the power of how we should go beyond those what we have traditionally been doing last decade, and therefore again, as Dr. Pendarkar sir completely highlighted it, that we have to go comprehensive. Therefore, only we'll able to identify something, and then we will be able to personalize the treatment to the patients. So we do not know, there may be beyond EGFR what we have been seeing beyond our NTRK, ROS, KRAS. We have seen other mutations are being identified in lung cancer. And I personally feel yes, we have to go beyond those 12 genes and possibly a highly comprehensive panel, not 52 gene panel. It should be something like more than 300 gene panel, 300 to as we have 400, 500, 600, and even more gene panel. Those should be used here. Yes, provided the cost is it is affordable.
So rightly said, and also I feel because tissue sometimes becoming, if you do one test at a time, you are not able to, so you are going to exhaust your tissue, which is very limited, specifically in lung cancer, where there are small biopsy specimens. So having a broader approach is always going to be helpful. So we move to the last part of the section, I think. So what is, as I mentioned, is the tissue remains an issue. So there, how often all of you face these issues with respect to failure on the solid tumor, particularly with the solid tumor. So how often you face these issues where the test has failed? Dr. Shafi, if you could.
I think yes, tissue is an issue, and particularly there, what we have now heard terminology, Indian FFP, because what we have seen that every laboratory has their own way of preparing FFP blocks, and now those labs who have come up with NGS testing, they optimize their protocols based on maybe one or two laboratories where they get the FFP blocks, and then they get the tissue blocks from other laboratories, and which have been completely prepared a different way, and there is a high chances of they fail. So what we see is, in the literature, what is my experience, about up to 25 to 30% of tissue actually fail in NGS testing, because either the low quality of DNA or low amount of DNA is there, or in sometimes we see just VEX is there, there's no tumor content. And if there is a tumor content, the content of tumor is so low that it becomes very difficult to actually capture what is happening in the tumor, because we know tumor is heterogeneous, and therefore you don't get complete picture. So I personally feel now we should move to the liquid biopsy. Liquid biopsy, we know now technology has improved drastically and have the ability to get the NGS sequencing done even as low as 1 nanogram of input DNA, and we get more than 1 nanogram of in DNA from a liquid biopsy, and we should be able to do the testing on a liquid biopsy.
Yeah. So I think you very rightly pointed out. I just wanted to bring out this thing because you can to everyone that your NGS can fail, and particularly in solid tumors, and around 20 to 25% of the cases it fails. And primarily, one of the key reasons that we have looked at is because of the fixation or how we prepare our block in the laboratory. So each one of us who is sending a block or a tissue should look into that the blocks are processed in the right way, because if they are not done or fixed in, over fixation also destroys it. Under fixation definitely doesn't help anyone in the molecular laboratory. So one of the things that we need to do in the laboratory also as a clinician also, to see that the blocks are prepared in the right way, and that's how we can get the results. So last question to everyone, once again, Dr. Shafi has already highlighted the comments regarding the role of liquid biopsy in your practice. Are you currently using liquid biopsy in your practice? Dr. Arvind, if you can just comment on that.
Yeah, I'm there. Yes, I think, liquid biopsies, definitely, at least when we are discussing in lung, at least in the recurrent settings, at times, I think there is a there is a strong consideration to consider liquid biopsies. But I know there have been some centers who have started actually treating based on liquid biopsies itself. Maybe that's something there in the future. The concordance seems to be 80% higher, or depending upon what study you go. But as you said, in lung cancers, tissue becomes an issue. So it is in those cancers in which I think there is a slightly more consideration, because some of our patients are slightly poor performance status to access the tissue for these patients by, say, a percutaneous or any other route becomes a challenge. And in which cases, I think in clinical practice, we have been forced to sort of take the liquid biopsy route at diagnosis for some patients. But definitely when it comes to the recurrence, to look for the resistant patterns, we have relied on liquid biopsies, and definitely for EGFR and many of the markers for lung. I think this is one area where we currently are using liquid biopsies in our clinical practice.
So do you use circulating tumor cells or cell-free DNA? So you any comments on that?
No, we use both essentially, but as I said, like, the sort of, the usage is far and few. We right now, of course, we know that this technology is there available, but somehow I think there is some sort of a consensus to obtain some tumor as much as possible, and in cases where the obtaining tumor becomes a challenge, or patients refusing, or for any other reasons, I think we go for either of the technologies.
Dr. Shafi, you would like to comment on anything with respect to CTCs specifically?
Yes, CTCs have, lately it has become very important, and particularly, as an MRD tool, where you can use the CTCs at regular intervals to measure that, if there is a disease is completely disappeared. And we have seen that, when PET scan has not detected anything, but CTCs were present, and then MRI was done, and then MRI has confirmed that, there's an tumor lesions there, and therefore it becomes important. So, and again, we have to see, it has to be carefully used, when and how we utilize it.
So with that, I come to the end of this session. So thank you so much, the panelists, and thank you ISO and Dr. Richa and Dr. Damodar for this opportunity. I hand over the desk to you. Thank you.
Thank you, Dr. Neeraj, and all the esteemed panelists. That was really a very informative and educative session. So I now request our chairperson, Dr. J.K. Singh sir and Dr. Sanjeev Mishra sir, to have final comment the session. I think they have left. Okay. So before we conclude, I think we have one or two questions left. One is regarding the availability of MSI testing in Northeast India. So I think availability is also an issue.
I think nowadays availability, it is available with most laboratories. It can be tested both, as I showed, both by a IHC, both by PCR based testing in the lab, and also by next generation sequencing with various comprehensive approaches being available. As far as I am aware, there are many labs which are offering that kind of services.
Okay. So I think Dr. J.K. Singh sir is there for his comments.
I think as far as the testing is concerned, he's right that it's not now a problem. You can have the test done in any good lab, and they will give you the result within the shortest time. So testing is never a problem. Now the availability and the cost of the drugs is very, very important. And whatever the drugs we have been using, it's very costly, and even the government is not coming to help the poor patient in a big way. They are not coming. Only a small subset of patients, they are able to have the drug to be taken. Otherwise, for mostly the patient, that Dr. Richa, that patients are coming from the village areas. Most of the patients we are getting from the rural areas also. We are not in metro town. So the availability of the drug is never a problem. Only thing, cost of the drug is a problem. If the cost can be minimized, I think definitely it will be a great effect, because molecular is the future, and that will certainly change the scenario of the cancer in the future, what I'm thinking. So I'm sure if the government should take up, and if our association, ISO and other associations should approach to the government regarding the cost of the drug to be minimized, I think it will be great help to the poor patient. Thank you.
Thank you, sir, for your comments. So any more comments? Okay. So I take this opportunity, thank all the faculties, all the speakers, all the panelists. So I think it was a very informative session. Now we know how to select the sample, how to select the approach, how to interpret the result. And I suppose that although many challenges still remain, the information which we have gained from, high throughput technologies like the NGS, they are really laying the foundation for personalized treatment for our patients. And with this, I invite Dr. Arvind Krishnamurthy, Secretary ISO, for his vote of thanks.
Yeah, thank you. I think it's an honor to sort of to give the vote of thanks, and I'm so happy that such a niche program is being conducted under the umbrella of the ISO. And I must immensely thank Dr. Richa and Dr. Damodar Gupta for sort of spearheading it and getting a wonderful panel of speakers as well as researchers. I think the science of oncology has evolved in a big way. I remember that when we were training, it was a one-size-fit-all approach. There was no concept of personalized medicine, and I remember a lot of patients who came with stage 4 cancers. A lot of them were subjected to supportive care itself. Sometimes even it was based on clinical radiological grounds. But now we know the immense power of precision medicine. And we do have, at least in some cancers, for example, lung cancers, with stage 4 also, we have patients living for many years, 10, 11 years also for stage 4 lung cancers, which is something that is what's unheard of just about 10, 15 years prior. So we have made leaps and bounds. And I'm happy that, the panel focused on comprehensive genomic profiling, next-gen sequencing, not just its implications are very many, right from prevention to early diagnosis, diagnostics, therapeutics as well, a choice of treatments, mechanisms of resistance, all those things. And as was rightly pointed out, I think costs were an issue, but I feel that the costs of sequencing are coming down. And I believe that the cost will further come down. There are good Indian players as well. And I'm happy to say that, some of their reports match that of the traditional people who have been there. So I think now we have multiple options, and I'm sure that the costs will come down. And also access to remote centers also, I think they have places in which you can sort of ship these samples to some of these centers, and they're able to give the report. So now what's happening is we have a wealth of data with us. Every oncologist has a wealth of data, and it is very, very important for us how to mine the data, and that is where the importance of molecular tumor boards was there. And Dr. Neeraj who Arora really elegantly pointed out some of the case capsules, which I was keenly attending to. And I must thank all these other speakers as well. Dr. Pendarkar, Dr. Damodar Gupta also, and Dr. Shafi also for their powerful insights. So right now, what is important is for us to unlearn certain things and relearn certain things. And I'm happy to say that in the ISO banner, we could sort of start off with a niche program like this. And I'm sure that many such academic events would happen in the future. I must also at this point tell you that we parallelly we had an ICC 2022 meeting also in the initial part. That's why I could join late. I would urge everybody to sort of strongly register for the Indian Cancer Congress. I think that is one big meeting that we will be having. Of course, we will be having a lot more of such hybrid meetings under the ISO banner in the near future as well. We will keep everybody updated of that. But I'm sure that all our eyes are trained to the Indian Cancer Congress. I would encourage everybody to register, those who have not registered, and encourage all your colleagues to register. And as an Indian Society of Oncology body, we will be heavily represented in the ICC. And definitely, I'm sure that we will all meet in hybrid forums and occasional physical forums in between. But for a major ISO banner meeting, we would have to wait for the ICC. And in case you've got any other suggestions for the scientific program which we are making for the ICC, I would urge everybody to sort of reach out to us or Dr. Bilimaga or any of the senior past presidents and office bearers of the ISO. And we would be keen to sort of have this topic also sort of included in some form in the scientific program of the Indian Cancer Congress. So, once again,
Dr. Arvind, just with respect to the ICC, just would mention I am the secretary for the Molecular Pathology Association of India, and we also will be participating in the ICC Cancer Congress that will be held. So, a lot of I think what you pointed out, strong presence in all areas which can benefit each one of us in different ways. So, thank you for bringing that out.
Great, great. I think looking forward for your inputs. And I think you all have a greater responsibility of educating all our oncologists. And I'm so happy that you, Dr. Gawar, and all the other people, major Indian players, now you have sort of created a sort of a benchmark, and we look up to you to sort of provide us insights into the tumor boards of molecular tumor boards, which is becoming a very, very integral part of. I'm sure that all the tumor boards will have a molecular biologist or oncologist in the future. And really hats off to you once again, Richa and Damodar, for bringing up a good program. And I would like to thank you all for the opportunity to listen to you all. Thank you once again.
Thank you. Thank you.
Thank you.
Thank you, sir. And thank you,
so
ISO,
much.
for providing us this platform to bring the clinicians and the researchers together. And I really like the idea of what Dinesh sir said. We need to archive. If we can have an archive of, every clinician, every researchers, they have their own wealth of information. And if we can bring them together and the ISO can take it forward, it will be a big thing. So thank you. Dr. Damodar, would you like to give?
Thank you so much. I think everything is shared by each one of us. Archiving and keeping those things as per the regulatory norm is one of the most required thing for all the clinician. Either it is today or tomorrow, we'll have to relook into all those things. Even if samples are preserved or available in storage condition, we can relook on all those samples through a particular channel, and then again look for the precision medicine or personalized medicine or for a particular kind of cancer or different type of cancers. Yeah, thank you so much.
So with this, I thank Rajesh and his team for organizing this event. So can we just close it?
Thank you. Thank you, everyone.
Thank you, everyone.
See you. Bye. Thank you.
Thank you, Dr.